Hello, everybody. My name is Sarah Kremen, and I wanted to welcome you all to our November 2020 UCLA Dementia Colloquium. We are very excited today to have our guest, Dr. Meyer, talking to us today about the gut microbiome and the relationship to the brain, which is definitely a hot topic and something I know that we are all very interested in. I just wanted to introduce Dr. Meyer. He received his MD PhD degree from Ludwig Maximilians University in Munich, Germany, and completed his residency at Vancouver General Hospital in Vancouver, Canada, and his GI fellowship at the UCLA VA Wadsworth Training Program. He has a long career interest in clinical and research aspects of brain-body interactions with a long-standing focus on the bidirectional communication between the brain and the gut in health and disease. He is recognized as one of the leading investigators in the world of brain-gut microbiome interactions in GI disorders, including functional and inflammatory bowel disorders and obesity. And he has been funded by the National Institutes of Health since 1989. We're really excited to hear what he has to say today, particularly because this topic has become one of the hot topics in the world of neurodegeneration. So thanks very much for talking with us today.
Yes, thanks very much, Sarah, for inviting me. I know this has been sort of difficult because of the pandemic situation, but I'm delighted to give this talk today. Just as a couple of words of introduction, why a gastroenterologist like myself would give a talk like this. I still ask myself sometimes this, but so the brief explanation is that I had a career-long interest in brain-gut interactions at a time when this was definitely not a catchword popular with the lay public and gastroenterology. And then about 10 years ago, and this was focused really because this were the only diseases where there was any obvious rationale to apply this research area. I studied primarily gastrointestinal diseases, mainly functional GI disorders like IBS, but also inflammatory bowel disease. And then about 10 years ago, with the explosion of microbiome science and the focus on the gut microbiome, we clearly evaluated the relevance of the microbiome to our models and understanding. We always thought that what we knew about the brain-gut interactions 10 years ago was sufficient to explain pretty much the interactions between the mind and the gut and these diseases. But, you know, we were quite wrong about this. So for the last 10 years, this has, the brain-gut microbiome axis has become the main focus. And even though I'm still funded on, by the NIH for our IBS research, my interest is really broadened significantly to now include a variety of brain disorders, including cognitive decline, Parkinson's disease, ingestive behavior, obesity, and autism spectrum. Not that I'm an expert in all or any of these disorders, but there is definitely a niche that has opened up looking at the interactions of the gut microbiome with the brain-gut axis in the development and chronicity of these diseases. So just as a brief explanation why I'm here today to give this talk to this, I imagine, mainly neurological audience. So just a brief disclosure, even though, these companies are all sort of working in this brain-gut microbiome space, I don't have any real conflict in terms of this particular talk. I've broken down my presentation into three areas. The sort of a general brain-gut microbiome system 101. I should also mention, so we used to call this the brain-gut microbiome axis. I think since we have sort of moved really towards a systems biological understanding of these bidirectional interactions, I think system is more appropriate. Then I'll I'll talk about some of the communication channels that exist between the periphery and the brain within the system, and then in the third part, address the potential role or the evidence that supports the role of alterations in brain-gut microbiome interactions in cognitive decline. As you probably all know, this has been not just in the scientific literature, but also in the lay press, based on amazing preclinical studies performed in mainly germ-free mice initially. There's been a tremendous enthusiasm, and I would say it's gone way beyond the data in terms of implications for human disease, depression, anxiety, and you can see their cognitive decline. Alzheimer's disease is one of those disorders that has been implicated early based on findings in animal models. So for some of these disorders, science is confirming, now human studies are confirming that the initial assumptions and concepts based on animal models were correct. For others, it's less obvious yet. So the original function that when people looked at the microbes largely in the colon was not really that, didn't seem that relevant really for brain science as a neuroscience. It had a lot to do with metabolism, for example, resistant starch or plant-based fiber breaking down into short-chain fatty acids, or lignans into phytoestrogens, an interesting one, xenobiotics into multiple metabolites. So the conversion of many drugs, possibly the majority of the drugs that we take, are being converted by the microbes, in addition to what the liver does to drug metabolism. And then what's become obvious that many of these metabolites have relevant functions beyond the gut, and that's shown on the right side. So pretty much all specialties have identified important influences of some of the metabolites or some of the changes in the gut environment, like low-grade inflammation. So the immune system was an early one that similar to the cardiovascular system, some pretty strong data has come out of that certain risk factor play a role in cardiovascular disease. Risk factor dependent on gut microbial metabolism. Gastroenterologists have function on the enteric nervous system. Metabolism has become a big area, and brain and behavior and the stress axis have sort of been pretty much become our main interest in our group. But this transition from this original view and the gut microbiome being a second liver or an additional organ to one that is really involved in the regulation of many organ and systems functions in the body has really happened in the last 10 years, I would say. And so this is kind of a systems biological rendering of these interactions that sort of emerged in the last few years. A key point is that there's really three players. There's the gut microbiota, and the brain. The interactions between these three players are bidirectional, really important concept. So the brain can influence many of the gut functions, and this is something that I studied early in my career, brain-gut interactions. The brain can also directly modulate gut microbial function and relative abundances. And then there's obviously a very close interaction between the gut microbes and the gut of the host. And so many of these molecules, we know these classes of molecules that are involved, and I'll get into this into greater detail. But I'm showing this at the beginning because it illustrates that to study and understand these systems, you need a concept that goes beyond a a simple linear cause and effect model. I think it's really a systems biological challenge to identify what these bidirectional interactions and feedback loops, how this ultimately determines the function, the overall function of that system. Should also say this is an ancient system in evolutionary terms. Some of the most primitive marine animals, like the hydra, already had were basically floating digestive tubes with a tube surrounded by a nerve net, sort of the predecessor of the enteric nervous system and then the central nervous system. And at some point, certain algae settled inside of that floating tube and started communicating with these nerve nets, and with lateral exchange of gene material. That's really could be considered the origin of development of evolution of neurotransmitters and signaling molecules that have been maintained up to this date. So very similar molecules are produced by the microbes, cells within our gut, including the enteric nervous system, and within the brain. So let me first show something how this has been a certain evolution in this field in terms of our understanding. So initially the focus was on relative abundances of gut microbes. It's a technique 16S RNA sequencing. It's fairly straightforward and inexpensive. So a lot of studies have been done using that technique. And this is a sort of a good example of how the early concepts and papers were written that a particular organism is involved in the increased production of a particular signaling molecules. This case, a neuromodulator, oxytocin. And then in mouse models, in vitro models, this has been shown to regulate neuronal plasticity, but also behavior in animal models. So this connection has been established in mouse models of social behavior and brain function. For another strain of lactobacillus, and also very important, these are not, each species has often hundreds of strains, and these strains may do very different things. So in this case, so another lactobacillus strain has been shown to be associated with an increased production of GABA and signaling through the vagus nerve to the brain with a reduction of stress responsiveness, reduction of anxiety, and increased vagal mesenteric firing. Bifidobacterium longum, another one, BDNF, Bacteroides, Sarkis Mosmanian, and Elaine Chow have sort of done the seminal experiments, have identified this particular metabolite that plays a role in anxiety-like and autism-like behaviors, altered communication. And then short-chain fatty acid producing bacteria, there's a lot of those. Short-chain fatty acids are probably some of the best studied. Signaling molecules mainly have positive and beneficial effect on, not just on the gut, like for example, permeability, on mucosal growth, but also beyond the gut on nerves and on the brain. And here's some of the evidence, similar to what I said earlier, can be in both directions. In one model, in a Parkinson's-like animal model, also from Sarkis Mosmanian's group, it's been shown that increased short-chain fatty acids lead to activation of microglia that may play a role in this autism-like behavior. So they made in the Parkinson's-like behavior that makes these symptoms in animals worse. But in general, I would say the concept is that these are some of the most beneficial molecules that microbes produce, mainly from plant-based fiber. So, but the field is a lot more complicated. That's what I said earlier. So the previous slide showed you a pretty linear correlation between a particular microbial species or strain, particular behavior, and a particular neurological brain change. It's a lot more complicated, as is obvious when we just look at numbers. So there's not only, several trillion microbes in our gut, but most importantly, there are millions of microbial genes, really outnumbering our human genes. You can see that the human genes in this schematic from Rob Knight showed the red line around the body contour, whereas all the rest would be occupied quantitatively by microbial genes. What's also fascinating that all the textbooks up to now have been written ignoring 99% of this whole genome, this hologenome system, and so maybe not. It's probably not a coincidence that there's a lot of chronic diseases that we don't really understand and don't have therapies for, because we neglected a big part of the information that's present in our bodies in conjunction with the microbes. So this takes a much more complicated, a much more sophisticated and systems-oriented approach. Complicating this is also that we only know a small fraction of the function of these genes. So we're just really scratching on the surface of what this really encodes, what functions, and what it means for our health and our diseases. This shows you sort of quite different from the early rendering that I showed you. So there's hundreds of thousands of metabolites that are generated based on this complex genome, microbial genome. And many of these metabolites interact with each other. It's not that one metabolite causes one end result in the body, either in the brain or in the immune system. They interact with each other, and they can be produced. So the same metabolite can be produced by different microbial strains or species. So just knowing the microbe doesn't really tell you what metabolites are being generated, what the functionality is. What's also really important is that one major impact in the adult that we currently can study, because we use a sort of a systems approach with diet. There's so many things in the diet, tens of thousands of phytochemicals, for example, or different fiber molecules that all interact with different microbes, generating different microbial metabolic functions that then have an influence on the whole organism. So going from this sort of intimidating complexity to how can we sort of bring some order into this? So the metabolites can generally be broken down into diet derived, as I just mentioned. So these are amino acids play a big role because they feed the microbes, and the microbes generate metabolites by metabolizing them. Polysaccharides, particularly complex polysaccharides or sugars, carbohydrates that are not absorbed intact in the small intestine, but that require enzymes, that require microbial enzymes for metabolism. And polyphenols, another group, probably the least studied to date. Most of this has focused really on the fiber molecules, amino acids. But polyphenols are another amazing group of molecules. Thousands of these large molecules are known. They're generally marketed as supplements of antioxidant supplements. And that misnomer comes from the fact that they have been studied in vitro on cell cultures. The cells don't have to worry about actually metabolizing these molecules. But if you ingest those molecules, they cannot be absorbed by the small intestine, but move down into the distal ileum and into the colon, where they're metabolized into small chemical entities, which then act locally and act on systemically all the way up to the brain. So diet-derived metabolites, host-derived metabolites, really important component. Primary bile acids, and I'll come back to this in the context of cognitive decline in Alzheimer's disease, also estrogens and steroid hormones. There are these circular processes that these molecules synthesized in the liver are excreted into the bile, into the gut, are then metabolized by the microbes, and then reabsorbed into the systemic circulation. So depending on the microbial composition, the plasma levels of not just primary bile acids, but also estrogen and other steroid hormones are dependent on the relative abundances and diversity of the gut microbiome. And then there's de novo synthesis. Remember the 20 million genes of biosynthetic precursors that have also, it can either be toxins or can be signaling molecules. Oops. Now, this field is rapidly moving forward to identify which of these molecules are actually relevant in modulating host physiology. So this technique of gut microbiota metabolome bioactivity screening in the pipeline, and then matching this with a G protein coupled receptor library, one can identify microbial metabolites that match some of these receptors, and then this gives an implication, depending on where these receptors are expressed, to which target these microbes signal. And this is sort of shown here from the food or medications that we take. The microbes metabolite those and then generate molecules that act on G protein coupled receptors. So it's a translational process from food-related metabolites into neurotransmitters that can act on G protein coupled receptors. Now let me go through a couple of these communication channels that have been identified by which the microbes can communicate with the brain. This is always a bidirectional communication, as I mentioned earlier, but I'll show a few examples from the gut up to the brain. So this is a rat brain. I'm glad in this review they used the rat brain. Typically, many review articles have always put a human brain there, implicating the same thing happens in humans. We don't really know this for a lot of these mechanisms. And this is going top down from the gut being on top to the brain. So vagus nerve modulation has been implicated for a lot of the signaling. And this comes from the fact that if you have a mouse model and you do an intervention targeted at the gut microbiome, it changes the behavior of this mouse. And then you do a vagotomy, you no longer see the behavior, so it starts to be vagally mediated, which is probably true for a lot of these molecules. And it's just shown here that certain, certain metabolites would activate vagal afferent neurons and generate both an altered behavior, but also often neuroplastic changes. And just mentioned this a couple of minutes ago, we are now able to identify G protein coupled receptors from these libraries. So now it's not just as came, the message came out from these early experiments, you cut the vagus nerve and you say it's vagally mediated. Well, the vagus is not a homogeneous pathway. It's a very complicated communication system. First of all, it's bidirectional. It goes from the top down to the gut. But also the bottom-up pathways are very heterogeneous, with different G protein coupled receptors being expressed on different subtypes of these fibers. So, but it gives an idea how in the future, and some investigators like Elaine Chow's group and one of her postdocs are actually working on this system. In the near future, we'll be able to really link certain microbial metabolites to subtypes of the signaling molecules that get to the brain. What's obviously another huge question is how is this information then integrated in the nodose ganglia and the nucleus tractus solitarius before it reaches cortical regions within the brain and emotional circuits within the brain. Another well-studied communication system is from enterochromaffin cells in the gut to the brain. And I'll show you this in a minute in greater detail. These enterochromaffin cells and enteroendocrine cells are cells that are interspersed in the lining of the gut with epithelial cells. And they contain, in the case of enterochromaffin cells, serotonin. In the case of enteroendocrine cells, gut hormones that mediate satiety or appetite. And when these cells, so there was a lot of literature before the gut microbiome came even on the horizon. And this was a big part of enteric neurobiology, that how these cells can modulate various functions, both of the gut, but also the brain. Now we know, so here are these cells interspersed within the epithelial layer. If you put all these cells together, it would be the biggest endocrine organ of our body. And the gut microbes used some of their metabolites, such as secondary bile acids, short-chain fatty acids, amino acid metabolites, to activate receptors on these cells that then lead to both an increase in the synthesis and in the release of serotonin, in the case of the enteroendocrine cells, of other hormones, gut hormones, which are then thought to move primarily through the vagus nerve to the brain. And I'll show you this in a little bit more detail, again showing you the complexity of this field. So tryptophan is at the beginning of these signaling pathways. Here's the brain connected to one of these enterochromaffin cells. They're unique cells. They're not just the simple endocrine cells, but they have these lateral extensions called neuropods that form synapses with afferent pathways. Certainly the afferent vagal pathways. They're also modulated top down by the autonomic nervous system in their excitability. But again, from Elaine Shaw's work, there's this bidirectional interaction between a group of spore-forming Clostridiales microbes that use their metabolites, short-chain fatty acids, and secondary bile acids to activate these cells and stimulate an enzyme, tryptophan hydroxylase, which is the rate-limiting enzyme for serotonin synthesis, and also can lead to the release of serotonin then onto these synapses. We now also know this is not just a one-directional pathway, but it's the serotonin released from these cells acts on certain certain groups of these microbes, and is taken up. These microbes have a mechanism similar to the serotonin reuptake mechanism on neurons and platelets. So there is really a bidirectional circle that a certain group of microbes, mainly Clostridiales, are in very close synchrony with host cells that have a directly link to the brain. So serotonin is modulated by gut microbes. Kynurenine, another tryptophan metabolite, is also the synthesis is highly modulated by, for example, by lactobacilli, who can influence this conversion of tryptophan to kynurenine. I won't get into the detail, but kynurenine has been mainly shown to have negative effects on the brain, on the nervous system. We'll come back to this as well. So the ratio between tryptophan and kynurenine has often been used as an indicator for various brain disorders involved in neuroinflammation. And then, so both of these enzymes that are involved here are host enzymes. In contrast, indoles, the third pathway of tryptophan, is a specific gut microbial mechanism, an enzyme, by which gut microbes can convert tryptophan into indoles, and then a variety of other metabolites, indoxyl sulfate. And again, just like kynurenine, some of these indole metabolites have been implicated in not just one, but several brain disorders, from Alzheimer's disease, cognitive decline, Parkinson's disease, autism spectrum disorders. So some of these mechanisms seem to be shared by several brain diseases. It's not just that one mechanism or one molecule is specific for one disorder, but it's that there's a shared shared group of molecules that play a role in several of these brain gut disorders. So immune system modulation, another really important part. This again has made it into the lay press. So low grade gut inflammation has been associated with the typical Western diet, but also with chronic stress. And this low-grade immune activation has an influence on the nervous system. I'll show this in greater detail in just a minute. So, and finally, I mentioned earlier the polyphenols. The polyphenols also are the least understood, actually, in terms of their interactions with the brain. Annie Gupta in our group is involved in a application for an NIH study on this mechanism. But based on some animal models, this seems to be an important mechanism, again, in modulating glial cells and astrocytes within the brain. And you can imagine, so, in general, this has always been, these polyphenols have been thought to be just acting as simple antioxidants on individual cells. It's a lot more complicated. They have to be processed by the gut microbiome and then have very specific functions, either by themselves or in interaction with each other, on things like the blood-brain barrier and, for example, plaque formation within the brain. There is growing evidence that polyphenols and the diet rich in polyphenols, such as largely plant-based diet, have a modulating effect on cognitive decline and Alzheimer's disease. So here's the mechanism that we currently understand, how diet would relate into this low-grade inflammation. This is a healthy gut, high diversity of the gut microbiome, a thick and dense mucus layer that separates the microbes from the intestinal epithelium, and particularly from certain immune cells that have these extensions into this mucus layer, the dendritic cells. So in a healthy gut, there's very little of this activation of these cells. Now, with the typical Western diet, but also with chronic stress, the effects are quite similar. There's a decrease in microbes that produce the mucus layer that stimulate certain cells in the colon that then increase mucus production. So there's a decreased mucus thickness, this now allows certain microbes to come in contact, and these are not pathogenics. These are commensals in contact with the dendritic cells, leading to a low-grade immune activation in the gut. This can stay in the gut, or it can lead to a loosening of the tight junctions between epithelial cells. And so certain microbes can actually get through to the immune cells in the gut-based immune system. And this can spread beyond the gut. So a condition called metabolic endotoxemia, where you have an increase of inflammatory mediators in the systemic circulation, without an infection, neither gut infection or any other infection. And this has been associated in animal models with neuroinflammation. It's thought that this condition, which is probably present, this condition is probably present in a large number of people with the metabolic syndrome. So just imagine the numbers of how many people in the US have that condition. So how many people have also neuroinflammation. But the genetic risk, underlying genetic risk in the individual ultimately determines if that person comes down with any of the diseases. So not everybody who has this or even this will develop any of these diseases. So clearly genetic predisposition, in addition to this common process, it's possible that up to 40% of the US population suffer from this, certainly at increased risk for that. It's also been speculated recently with the COVID-19 situation that this process may play a role in the increased susceptibility of patients with comorbid disorders, because there's depression, there's metabolic syndrome, and other, so-called comorbid disorder, cardiovascular disease, that this process even plays a role in the increased vulnerability to severe COVID-19 infections and clinical courses. So I told you a lot about the mechanisms. So coming back to the diseases, so this has been the history of this field: neurological mouse models showing gut dysbiosis. So there's mouse models for all of these diseases. So depending where you're coming from, you would say these are very realistic models. Others, you question as a clinical investigator, I often wonder, seeing the complexity of patients with any of these diseases, if these models are really applicable and translatable. But a lot of the early evidence came from these kind of studies. So this has gone to another stage. So now, so there's studies with fecal transplants. I think this is sort of the main progress that's been happening in the field, that investigators have taken fecal material from a well-characterized patient population, put them into germ-free humanized mice, and then look at if they reproduce some of these behaviors or these mouse models. It's also been shown that broad-spectrum antibiotic treatment have a modulating effect, and similar probiotic interventions. So probiotics do a lot in these animal models, have a lot of beneficial effects, but this has been very difficult to translate into humans. And then we have the last stage, that quite a few neurological disorders now that have been shown to have a dysregulated gut microbiome, so-called gut dysbiosis. But these are cross-sectional associational studies. We don't know what's the cause and effect. It could be that the stress that patients with some of these disorders experience modulate the gut microbiome, or it can be that that certain microbial metabolites really play a role. And this is made particularly complicated because you can't do this in humans, while given the circular interaction, the systems properties of many of these interactions. But there certainly has been progress, I would say, from here to here in the last 10 years, and it's been accelerated. So I think we are definitely getting closer in identifying specific roles for the gut microbiome in neurological diseases. I mentioned this several times, this bidirectional interaction, and this is important. There's actually more evidence, more early evidence, in preceding the current explosion of data about bottom-up signaling, that stress, but also certain negative emotions can alter the gut microbiome, either indirectly by changing like regional motility, secretion, antimicrobial peptide secretion, but also directly by releasing a norepinephrine into the gut lumen, acting on adrenergic-like quorum sensing receptors on certain microbes, which then change the gene expression pattern and behavior of these microbes. And this is probably not limited to norepinephrine. It's probably a feature of all the endocrine cells in the gut that can release their content into the gut upon stress and modulate behavior of these microbes. Let's see, time-wise, yeah, I'm probably gonna, I just go through this very quickly, just this kind of related. So inflammation and neurodegeneration is implicated in psychiatry as well. And again, linking an unhealthy Western diet to an increased risk of many of these neural processes. This has been reviewed in a recent article. It's actually led to a whole new field of so-called nutritional psychiatry, that Western diet with its contents, so decreased amounts of polyphenols, increased amount of fat, decreased amounts of fiber, lead to a systemic low-grade inflammation, alteration in the gut microbial composition, the metabolites, in a change in this tryptophan kynurenine metabolism that I mentioned earlier, that has been shown to be a feature of many brain disorders. This study illustrates, or this group of studies illustrates where the field has gone to. So investigators have identified, going beyond cross-sectional studies, characterizing a group of individuals or phenotyping a group of individuals with a disease like major depressive disorders, looking at their relative abundance, differences in the relative abundance of gut microbes, which in this case you can see the red ones, the microbes that come from the disease population, separate quite clearly from the healthy controls. This is reflected also in the difference in the metabolite profile and in the relative abundances of microbes. Then fecal material from these patients is then put into germ-free humanized mouse models, which have been shown to show depression-like behavior if they got depression fecal transplant. And this mirrors then in the mouse feces the same separation between those mice that got the stool from the depressed patients, from the non-depressed patients, and is reflected in a very different metabolite profile. These kind of studies, this kind of approach has been done now in several human disorders, and I would say right now is the closest that we have gotten into getting into causality between microbes and altered human brain function. The model that has evolved, and it probably fits best, not just for depression, but for all the brain gut disorders, that alterations at the brain level, so in terms of depression, anhedonia, stress reactivity, there's an extensive literature obviously on this in psychiatry, leading to an increased chronic stress response, which modulates what's been called the gut connectome, all these cells present in the gut, including the microbes. And then the output of that altered system, mainly the metabolites from the modulated microbes, feed then back in form of their microbial neuroactive metabolites or immune mediators to the brain and reinforce this circular interaction. So we don't know for sure. This fits very well in depression and anxiety and obesity. How this applies to Alzheimer's disease, what early or primary brain changes might drive this, or if in Alzheimer's disease is actually starting at the gut level and changes the brain and then initiates the circle, is currently not known. Okay, so cognitive decline. We've been fortunate to been invited by Rima Kadurian from Duke University to participate in this U19 consortium with the leadership, Sarkis Masmanian and Rob Knight, which is a phenomenal consortium combining the leading centers in Europe and the US. And so we've been invited just on a very specific function of this to integrate gut microbial alterations and in metabolites with brain function. There's a lot of existing data on brain data, metabolites data, but also the plan on 10,000 or more new patients to study this prospectively. Chen Labis from our center is sort of the lead in this collaboration. It's been a lot of reports. So after all the things that I've told you about the brain-gut microbiome interaction, it's probably not surprising that there is now a rapidly growing literature on the potential role of these altered interactions in cognitive decline and Alzheimer's disease. I'll go through a couple of these studies listed here, but to summarize, what's sort of been emerging that chronic low-grade immune activation, as I showed you earlier, which may be diet-related, chronic stress-related, or a combination, will certainly plays a role in this low-grade neural activation in the brain glial cells, then an alteration in the tryptophan metabolites, both in the indoles, but also in the tryptophan ganurine ratio has been implicated as playing an important role. And the third one, which as a gastroenterologist, I thought, well, there's finally a molecule that I've been familiar with since the beginning of my career. Bile acids have also taken on a pretty interesting role in this. Won't have the time. I highly recommend this recent review article by Rima and Sarkis that have summarized the effects that have been reported on microbial generated compounds and molecules, both in terms of beneficial effects on neurons and glial cells, but also the ones that have been implicated in terms of detrimental effects, often related to inflammation, neurodegeneration. So most of them to inflammation, weakened blood-brain barrier. So this is sort of becoming clear that there's a group of molecules from microbes, so bad molecules from microbes that play a role in increasing the risk. And there's a group of molecules that are beneficial that not by coincidence are many of them sensitive to dietary interventions. Let's skip this. So in terms of bile acids, I'm sure there's somebody in the audience who is an expert, a better expert than me. The main thing, so bile acids, there's many bile acids, and they go just synthesized in the liver from cholesterol, to secrete into the bile as primary bile acids. And then here's the important step. So they're being dehydroxylated by a particular bacterial enzyme, this 7-alpha dehydroxylase, into a whole group of secondary bile acids. And what's been found in several studies now that the ratio of certain secondary bile acids to primary bile acids, shown here for this deoxygenol acid to primary bile acid. Here's for other ratios, that several of these ratios have an association with cognitive impairment and even with some of the biochemical markers of Alzheimer's disease. So this implicates, because this is happening in the gut and depends on which of these microbes express this, this enzyme, how abundant they are, how the diet influences the abundance of these particular enzymes. There's also genetic factors. This has all of a sudden put the gut microbes in a pretty central role in Alzheimer's disease and cognitive decline pathophysiology. Now, there's also now a small study, a handful of studies that looks at causal effects of an intervention. There's many studies, association studies, epidemiological studies that has shown that people on a Mediterranean diet, or which is basically a predominantly plant-based diet, have lower risks of Alzheimer's disease. But all these are association studies. And until recently, this is all we knew. It was pretty compelling. But this study by European Consortium showed that adherence to a Mediterranean diet led to an increased abundance of specific taxa that were positively associated with several markers of lower frailty and improved cognitive function, and negatively associated with inflammatory markers in the plasma, including C-reactive protein and some cytokines. And these microbiome changes were associated with also metabolic changes, increase in short chain fatty acid production, and again here, lower production of certain secondary bile acids that I just mentioned in the previous study, and other metabolites such as P-cresols, ethanol, and carbon dioxide. But this is really exciting that a dietary intervention would target actually a factor that has totally independently been identified as a risk factor. Now, what these investigators did, they took this to a whole new level. So they identified the most abundant microbial organisms that had a positive association with the diet, that increased and were associated with an improved function, and what they call the diet negative group. And the diet positive markers had an increased representation of certain species, including Fecalibacterium, Eubacterium, which are generally associated with healthy function, gut function. And diet negative markers were more abundant in another group of microbes. And then what they did is they used this ecosystem network analysis using graph theory, which some of you may use in your own brain studies. We use it in our own brain imaging studies as characterizing, quantifying interactions within brain networks. So they applied this to the gut microbiome community. And so without going into the details, so the diet positive are shown here in green. They showed greater, so measures of an influence of a particular nodes within a network, so degree centrality and betweenness centrality. So these healthy diet positive bacteria had a greater influence, were overrepresented in these diet positive group of microbes. And so they have pretty much identified a network structure at the gut level that correlates with behavioral functions. They didn't do brain imaging. We would love to do that kind of study correlating this gut microbial network, these network properties with the brain network properties, which hopefully we'll be able to do within this U19 consortium. But I think this is probably one of the most advanced studies that has shown both causality, but also a lot of mechanistic aspects of the interaction between diet, gut microbiome, metabolites, brain function, and symptoms of cognitive decline. So to conclude, sort of a whirlwind tour, but I hope I sort of showed you that there's an exponentially growing preclinical set of data supporting a role of the brain-gut microbiome axis in several brain disorders. This has really happened in the last 10 years. with increasing speed. This includes more recent evidence in neurodegenerative brain disorders, including cognitive decline and Alzheimer's disease. I think that U19 consortium is definitely one of the most promising ways of moving this field forward. Then cross-sectional, a lot of cross-sectional, but also now less than a handful of longitudinal human studies show role of an altered gut microbiome in the development of cognitive decline. And there's, somewhat promising studies that a dietary intervention has an effect both on the mechanisms, but also on the symptoms in Alzheimer's disease. And as I said, I think just the experience being part of that consortium together with Jen Levis has really convinced us that this is sort of a phenomenal synergistic ecological system now that works on different but interacting aspects of the brain-gut microbiome axis in Alzheimer's disease. And I didn't show you too many of our own primary data, but this is our Center for Neurobiology and Stress and Resilience program. And then if you want to read anything more on this topic, I recommend my first book, but also on my second book, which you can pre-order, which will come out in May of next year, which has a lot to do with the gut-immune interactions in disease. So thanks for your attention.